Good morning. This time around, we are going to turn our attention to a discussion of protein tertiary and quaternary structure with a specific emphasis on globular proteins. In our last recorded lecture, part 4, we talked about these features of fibrous proteins. But fibrous proteins and globular proteins are not shaped the same. They do not behave in the same way. So when we talk about protein structure, most commonly, we are going to be talking about protein structure as it relates to globular proteins. When we think about enzymes, they are by far, most commonly globular in structure. So when I say globular, I mean largely, approximately spherical in nature. So let me stop rambling. Let me just go ahead and jump right in. Before we talk about any specific cases of globular proteins, let's talk about some general features, big macroscopic features. So first, when we think about a globular protein, I want you to view this as a roughly spherical structure. Now, it's not just that it is roughly spherical. I want you to think about this kind of like the fruit Gushers candy. This may date me, this reference may date me, but Gushers, these are basically candy things that have a liquid in the interior. So when you bite into it, basically, the fruit juice gushes out. And maybe I shouldn't phrase that as being juice. Who knows what it is? It's definitely not good for you. But you have a distinct interior that is surrounded by a waxy exterior. Now, proteins being globular, the exterior is not going to be waxy, but the point is still the same, that you basically have a defined interior and a defined exterior. Now, when we talk about proteins, the exterior is going to be hydrophilic. It's interacting with the surrounding water. The interior of a protein is going to be mostly nonpolar material. This is due to the hydrophobic effect. So all of those nonpolar amino acids that go into building proteins, they're usually not going to be favorably interacting with the surrounding aqueous water. What's going to happen is protein structure is going to start forming such that those nonpolar amino acid side chains don't really interact with water. They get buried away in the protein interior. And what this does is it leads to a roughly spherical shape for the overall three-dimensional structure of the protein. And that three-dimensional shape is one that basically is a hydrophilic exterior and a nonpolar interior. So to go back to my Gushers example, basically if you took a Gusher, a single candy, and inverted it, you can see here the exterior is kind of waxy. You chew on this, and as you chew on it, the liquid interior just goes everywhere. If you flip this around and put the wax in the middle and made the exterior somehow liquid and solvent, polar, then you'd be good. You'd have yourself a mimic of a protein. Now, that doesn't really work so well when we talk about distributing candy, but whatever. The example doesn't always work. Okay, so for a globular protein, if you have a nonpolar interior and a hydrophilic exterior, what this suggests is that your amino acid side chains are distributed non-randomly. So when we look at nonpolar amino acids, so things like valine, leucine, isoleucine, methionine, proline, these occur mostly in the protein interior. They are kept in the protein interior away from contact with aqueous solvent. So remember, the hydrophobic effect, basically what we're seeing there is an entropic penalty is associated with the requirement for ordered water molecules around a nonpolar molecule. So here we minimize that entropic penalty. We make it so that waters don't have to become ordered by adopting a three-dimensional structure that puts all of your nonpolar stuff in the interior of the protein away from interaction with water. Now, other consequences include the fact that charged polar residues, so arginine, histine, lysine, aspartic acid, glutamic acid, these are usually located on the protein surface in contact with aqueous solvent, so water. So this is because the immersion of an ion in the virtually anhydrous, and by anhydrous I mean there's not much water in the protein interior, to put an ion in that protein interior would be energetically unfavorable. You don't want to put a charged ion in a nonpolar solvent. And in this case, when I say nonpolar solvent, what I mean is the protein interior, a nonpolar environment. An ion in that environment would not be thermodynamically favorable. When you do see, and you do sometimes see, these residues, arginine, histine, lysine, aspartic acid, glutamic acid, you do sometimes see them in the protein interior. And when I say sometimes, I mean very seldom, but you do sometimes see them there. When you do, they're going to be in very specific cases. They're either going to be hydrogen bonded to something else. And so imagine you have a lysine and an aspartic acid in the protein interior. They form hydrogen bonds in many cases when that happens in order to neutralize the charge that would be associated with those ions. Once you neutralize the charge, the thermodynamic penalty goes down significantly. Sometimes you will see secondary structures that are inserted into, say, a membrane environment, which is very nonpolar. In those cases, the hydrogen bonded nature of secondary structures make it such that you can neutralize the polarity and insert into a nonpolar environment. Okay, so there are other cases where you'll see these charged polar residues in the protein interior, and when you do, often you're going to see a perturbation to the pKa, the side chain pKa. So for arginine, for example, the guanidinium group has a pKa that is in the ballpark of about 12. Histidine has a pKa, the side chain does, of about 6. Lysine about 10. Aspartic and glutamic acid, roughly 3 to 4. So in those cases, you are going to see the pKa change to favor the neutral ionization state for each of these side chains when in the protein interior. That is, if there is no hydrogen bonding taking place to neutralize the charge. So for example, you might see the pKa's of arginine and lysine go down such that they become deprotonated and adopt a neutral ionization state. The pKa's of aspartic acid and glutamic acid may go up to make sure that they remain protonated and neutral in that environment. So pKa perturbations, or pKa tuning as it is sometimes called, this is a phenomenon that makes it so that you can actually put these residues in the protein interior. There are other residues that, think the uncharged polar groups like serine, threonine, asparagine, glutamine, and tyrosine. These are usually on the protein surface. They can hydrogen bond with water, but they can also occur in the protein interior. Again, their polarity is not terribly favored in terms of you putting a polar molecule into a nonpolar environment is not always the most favorable. When you do this, you're probably going to see some hydrogen bonding that is taking place to neutralize that charge. The first protein that we had a structure for was that of myoglobin. So myoglobin is an oxygen-binding protein found in all mammals. And over the years of 1957 to 1959, a man named John Kendrew and his colleagues worked on determining this structure. Now, before I actually tell you anything about it, I need to tell you a bit about the method of X-ray crystallography, which was the experimental approach that was used to determine this structure. Okay, let's take a moment and talk about the method of X-ray crystallography. The method itself, whether we're talking about proteins, small molecules, whatever the molecule type, what we're going to do is take a solution of that molecule and raise it to a very high concentration and try to encourage it to form crystals. So here, this is the general workflow. You'll see once you have your crystal, and whole careers are built upon protein crystallization. So don't think that this is something we're going to spend any significant time on here. But basically what you want your molecule to do is form a crystalline lattice, which I'll tell you about in a moment. And what you can do then is bombard your crystal with X-rays. All of the atoms, the electrons associated with atoms in your crystalline lattice, they're going to diffract those X-rays. And ultimately what you're going to do is have basically your crystal, and you're going to shoot it with X-rays, and on the other side of your crystal is going to be a detector. In the olden days, this was just a piece of photographic film, but X-rays are going to be scattered in very predictable ways. So the result is going to be that you're going to have on your film or your detector discrete spots. And these spots tell you something about where in the crystalline lattice each of your atoms is. And the spacing between spots tells you about the distance between the atoms. And so you can use basically the positioning of all of your spots on your film to build what's called an electron density map. So here, this is an example of your electron density map. This tells you where your electrons are, where your atoms are in the crystalline lattice. And then ultimately you can fit a protein into this. You have to do this usually one by one amino acid at a time, but you can fit in your residues such that ultimately you have an atomic resolution model. Now, let me walk through this for a moment. This is dependent upon you having a crystal where your proteins are packed in an ordered array. So here you can see different examples of protein crystals. Some of them are far prettier than others. These cubes are my favorite, but maybe these work as well. It's hard to tell from looking at the crystal whether you have an ordered crystalline array or something that is more disordered. So when we look at ordering, what we ultimately want are our proteins. So imagine I have one protein molecule here. I want it to pack in my crystalline lattice uniformly against all other protein molecules that are present. So as I hit my crystal with X-rays, what's going to happen is due to ordering, I'm going to see spots form on my detector or my film due to constructive interference. If you hit just one molecule with X-rays, your signal strength is going to be so weak that you're not actually going to see it. So if you have an ordered crystal, you get constructive interference. Imagine all of your protein molecules are scattering X-rays in exactly the same way. So if they are in phase, the scattered waves are in phase, you're going to see constructive interference and better signal. Now, sometimes you'll see what are called mosaic crystals. So proteins maybe pack in slightly different orientations. You'll see the squares that surround this one are slightly offset. So what can happen is your resolution goes down. Those spots that you saw in the film are not quite as discrete. Maybe they become a little smeary or your signal goes down. So if you see non-uniform packing in your crystal, your ultimate model, the resolution of your model is not going to be quite as good. Now here are two examples. So imagine this is my detector. I basically I have my protein crystal here in the middle. I'm hitting that with X-rays, which is why this part is basically sort of whited out. The intensity is so high here because that's where my laser beam is being shot. But on the back side here, you'll see the actual detector image. And you can see there are discrete spots here and there. And in this panel A view, these spots are not so well defined. So here, they're kind of smeary. You might have a spot here, but maybe it's just a smear. You see some defined spots out here, sure, but okay, this diffraction image probably came from a mosaic crystal where your protein was packed into a crystalline lattice, maybe in a not so uniform way. You'll notice that you don't get spots out here either, and that's due to destructive interference. So the more spots, the better. If you see spots way out here that's going to give you a much higher resolution model. So due to destructive interference, you don't really see more of the spots out here. Now, if you look at this image, you'll see beautiful data. So you'll see circles. So these circles tell you something about the symmetry of the protein in the crystal. That's very important if you want to determine the structure. But not only that, you see a lot of spots, and they are very well defined. So this would be an example of a well-ordered crystal that is ultimately going to yield very high resolution data. Now, this is important. The resolution is important because ultimately this is what you want. You want your atomic resolution model. But that is dependent upon the electron density map that you can build from those spots that I just showed you. So for a high resolution data set, you're going to be able to build your electron density map to be pretty nice. I mean, you can sort of follow this, and you can look at this mesh, for example, and say, oh, that looks like an arginine. Oh, cool. I mean, you can actually see what amino acid side chains should be there. Here, this looks a lot like a phenyl group of tyrosine. Cool. So that's what you want. And once you have this, you can build in your amino acids in a way that makes sense. And ultimately, what you're going to get by doing this is your atomic resolution model. Now, let's say you have poor resolution data. Maybe you get electron density that looks like this. So this is not great. This is not great at all. You don't really see any shape. If you try to fit in a polypeptide chain, you'll see your amino acid side chains don't really fit in with the electron density because the electron density is poor resolution. So in those cases, ultimately, you have to model in something that more or less lacks any side chain information. So here, this is a polyalanine alpha helix. And this was modeled because basically there was no other option. Now that we have discussed X-ray crystallography, we can transition back over to talking about myoglobin. So I'm showing you 3 panels here. The first is the X-ray diffraction pattern used, or one of the X-ray diffraction patterns used to determine that myoglobin structure. The next is the first structure of myoglobin that was reported in 1958. Okay, so what I want you to see here is that it's kind of blobby. I'll explain this more in a moment. It's kind of blobby. You can sort of see the overall structure, sure. And then flash forward to the 1960s, and now we have something that is much more high resolution. So this should show you something about the progression of the resolution of the model. The first X-ray structure of a protein, this one, myoglobin, was low resolution. This was due to low resolution crystals that gave sort of smeary data and limited the determination of an atomic resolution model. You still got some general features, but it wasn't as good as it could be. As they continued to work on it, they refined it such that you got better resolution, better diffraction, which is ultimately what led to this. Now let me give you a bit of history here. Why was this such a hard thing? Well, there are 2 big issues. The first of which is the source material. So John Kendrew had been working on this alongside his co-workers for quite a while. Over the years of 1957 to 1959, they intensely worked on the effort where they tried to determine a myoglobin structure. And Kendrew initially started by studying horse myoglobin, but he soon realized that the oxygen storing molecule in tissues should be more plentiful in aquatic mammals that dived and remained underwater for appreciable time periods. So here, if you think about this in the context of the method, I just explained X-ray crystallography to you, and I did this on purpose so that you would understand what it is I'm talking about. So our crystal itself, in order to get that, we have to raise our molecule to very high concentrations. And then we have to encourage it to in an ordered fashion, precipitate out of solution to form those crystals. This is a liquid to solid phase transition. Now, in order to do that, you need a protein solution that is of high concentration, which at the time required that you purify a protein that was present in an organism from an endogenous source present at high concentrations. Not all proteins are present in physiologic conditions at high enough concentrations to do this. So they reasoned, and correctly, that myoglobin, which is an oxygen transport protein, should be present in more plentiful quantities in diving mammals. So they looked at various diving mammals, think dolphins, whales, things like that, where you could readily get meat that was procured from these animals, and then you could purify myoglobin directly from that tissue. Okay, so they tried different types of whales. They thought whales might be a better way to go. They tried finback whale, sperm whale myoglobins. They were isolated and tried. Sperm whale myoglobin was actually selected because the myoglobin concentration was so high that the meat, sperm whale meat, was almost black. Now, myoglobin, it is an oxygen transport protein, but it has a metal present. So it has iron bound to it. So the iron gives it some color. Sperm whale meat was actually shipped. They got it from Peru, and they stored it frozen in large garbage cans in a deep freeze room down the hall. And the myoglobin was isolated and purified, and then used to grow protein crystals. This is a very interesting thing because these crystals grew so large that they actually had to be cut down to size. They were too large for traditional X-ray crystallography experiments, so they had to actually cut the crystals down to use them. But all of that work was not in vain. It led to this structure. Now, this is a low-resolution structure. You don't have the side chain information. It's just sort of blobby. Now, the reason I want to point out the blobbiness here, at the time, so this was over the years of 1957 to 59. This structure was actually 1958, but the effort spanned multiple years. At the time, we're looking at about 5 years after Watson and Crick report their model for DNA. Protein chemists did not initially accept this model. So think about this in context. Watson and Crick's model for DNA, it's a double helix, which is a pretty simple structure. Basically, you have a helix, and it just goes on. So here, what you should see is that this is not just a simple helix. There's, maybe something going on here. You can see in the refined structure that there's a helix, there's a little bend, there's another helix, there's some stuff. This is all in all alpha helical protein, but it's not as simple as the DNA double helix. So protein chemists at the time, they met this model with skepticism. The apparent lack of regularity in the structure of myoglobin was not immediately accepted. However, we know now that comparisons of over 100,000 protein structures, this has allowed us to really understand that proteins exhibit a remarkable degree of structural regularity. It's just that that structural regularity doesn't look like it did in DNA. Okay, now we can talk about the myoglobin structure itself. So here, I'm going to be working from this structure over here. And what I want you to know is that myoglobin, because it was the first protein structure to be reported ever, it is very well characterized and more or less serves as our paradigm for what it means to be a globular protein. So in the case of myoglobin, we have a protein that is 70% alpha helical. Now, I told you it's an all alpha helical protein. If you look at the structure, there's no beta composition here. But 70%, what about the other 30%? Well, you've got connectors. So you've got these connectors. these random coil structures that connect secondary structural features. So the other 30% are those connectors and other stuff. Most of the hydrophobic R groups for myoglobin are found in the protein interior. In fact, myoglobin, the structure, is a very compact one with a very, very hydrophobic interior that really only leaves room for 4 water molecules in that interior. So basically, as this thing forms its three-dimensional structure, it kicks out water, and what is left is a very compact, nonpolar, hydrophobic interior. Why do we care about this? Why do we care that the interior is compact and very hydrophobic? Well, this is actually quite important. It all centers on the iron. So if you look at this, you'll see there's an orange bit here in the middle. This is an iron atom. So we want this to be in the iron 2 form, the ferrous form. This is the form that binds oxygen. Remember, this is an oxygen binding protein. Myoglobin, what it does is it binds to oxygen and transports it through muscle tissues so that you can continue doing aerobic respiration. So this thing needs to have iron in the +2 state. Free heme groups in oxygenated solution, they become rapidly oxidized from the iron 2, the ferrous form, into the ferric iron 3 form. So upon oxidation, that iron does not bind oxygen. So this is an important thing to take note of, because the compact structure, though it doesn't readily appear that way here, what it does is it basically keeps this heme group, this is a heme group, it keeps it basically inaccessible, solvent inaccessible, makes it so that it doesn't interact with the surrounding oxygenated aqueous solution. So the likelihood that this iron is going to become oxidized from iron 2 to iron 3 is low as long as it is protected in the myoglobin interior. Now, this is, remember, important, because if that iron 2 is oxidized to iron 3, myoglobin can no longer do its job. It can't bind oxygen. It can't serve as an iron transport protein. So we need to make sure that the heme iron remains in that iron 2 state. So for those of you that have ever been to the grocery store, bought some meat, took it home, and maybe waited a little bit too long to cook your meat, you might have noticed that your ground beef starts to turn brown. What you're seeing there is the oxidation of that iron. It's not necessarily that there's anything wrong with the meat. It's just that your iron is becoming oxidized, and that changes the color of the meat. Remember the example of the sperm whale meat being almost black. That's due to the iron there. Okay, so it's a compact structure, very nonpolar interior, and there are also 4 proline residues that are present. So the 4th proline residue occurs within alpha helix, and you can actually see it right here. So the proline, proline is not all that happy to be in an alpha helix or any secondary structure, really. In fact, proline is generally referred to as a helix breaker or a helix terminator. When you see it adjacent to an alpha helix, it does something like this. It actually induces a kink that disrupts the regular structure of that alpha helix. Now we're going to look a little bit more in a little bit more depth here. Let me transition to the structure. Okay, let's look at this structure for myoglobin. Here I have pulled up the structure. If you are curious, you can go to the protein data bank and search accession code 1MBN. You'll find this structure. This is for myoglobin. Okay, so looking at this, I've highlighted a few features here. What I want you to see first is that there is no composition, no secondary structure composition here that is in any way in the beta conformation. This is, you've just got alpha helical segments, you've got rather random coiled structures here. I've also highlighted the prolines I was just talking about. So here I want to make note, here's one proline, and you'll see it actually occurs at the end of an alpha helix. Proline is often referred to as a helix terminator as well as a helix breaker. When found at the end of a helix, it's considered to be a helix terminator because it shows up here and disrupts that regular structure and basically terminates the helix from continuing. For this particular helix, if you look at the other end, you'll see another proline down here. Neither of these occur in the internal stretch of the alpha helix. They are basically sitting there flanking on either end that alpha helix as terminator elements to ensure that the helix spans basically the stretch, it goes as far as it needs to, and then we can stop it. Now, in this case, right here, you'll see we have an alpha helix, but there is sort of a change in direction of that alpha helix when you hit that proline. So proline, it really does not like to be in an alpha helix. When it is there, it induces these kinks, and in this case causes a 90-degree change in direction. All right, so looking at other features, here, if you look in the interior of the protein, there is a heme group. So this is the heme that I'm talking about. You can see here that at the center, in orange, there is an iron, and we'll talk more about the heme group in Chapter 5, but just at this point, know that blue equals nitrogen, orange equals iron, and all of the green stuff is carbon, and so let's just leave it there. So this heme group, it is held in place by interaction with the side chain of histidine. This is an imidazole. The imidazole has formed a coordination bond with the iron, and on the bottom side there is oxygen bound. So this is O2, and there's another histidine side chain, another imidazole on the bottom that helps to sort of position that oxygen. Now, remember, this has to be the iron 2 state. If this is the iron 3, this doesn't happen because iron 3 can't bind oxygen like iron 2 can. Now, I've also told you that this heme group is largely solvent inaccessible. So let me sort of give you a sense for what that looks like. So it doesn't really appear that way, but you'll see there is a cavity here. This is a surface view where the electrostatics are highlighted. There's a cavity in here, and it's, in fact, largely solvent inaccessible. Now, I want to highlight the charge distribution as well. You'll see on the surface there are patches of red and blue. Blue here represents positive charge, red here represents negative charge. So if I turn this around, you'll see there's a lot of charge on the surface of this protein, but if I zoom in here to where that heme group is, it becomes more whitish. So that white, this is, it tells you something about the charge. It's nonpolar in nature, so the interior here is largely nonpolar. All right, so this was a view of myoglobin. Now we can move back over to the discussion of protein structure. Okay, I just told you that myoglobin is a very compact protein. It adopts a three-dimensional structure wherein all of its constituent parts are packed into a very compact three-dimensional arrangement. Now this is interesting because if you compare to fibrous proteins, you'll see that the overall dimensions are very, very different. For example, if we look at a fibrous beta protein, or rather a fibrous structure built from a beta protein, for example, fibroin is a very common one. If you look at proteins involved in spider silk, they're all in the beta conformation, and because of that, that means that they're very extended. Remember, the beta secondary structure is a very extended conformation. In fact, the anti-parallel beta strand is stretched nearly as far, as long as a polypeptide can be stretched. So when we look at its dimensions, you'll see for the same number of amino acids in all three cases, the beta conformation would be about 2,000 angstroms long and 5 angstroms tall. So basically very long and skinny. Fibrous proteins built from the alpha helix, think alpha keratin. They're a little bit more compact due to the alpha helical secondary structure. Remember, the alpha helix is not as extended as the beta sheet is. So in that case, you're looking at 900 angstroms long, about 11 angstroms tall. Okay, cool. So what I've established to you so far is that fibrous proteins that are of the beta type are very long. Alpha helical proteins, also long, extended. But if we look at a native globular protein, this is quite a bit different. 100 angstroms in one direction, 60 angstroms tall. So the globular form is much, 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 much more compact than any fibrous protein that you're going to come across. Okay, so coming back to the heme for a moment, I wanted to just sort of give you another look at it. Well, the heme group in myoglobin, and you'll see this also when we talk about hemoglobin as well, it's going to be very solvent inaccessible. It is going to be built from an organic ring structure called a protoporphyrin. So this is that structure here. You'll see that there's a lot of conjugation, but there are essentially 4 nitrogens that wind up coordinating and holding the iron in place. If you look at the heme group from the side, it is planar, roughly planar, and the iron sits right at the middle. The imidazole side chain of histidine sits there above, coordinated with the iron, and oxygen below. So it is important, again, let me really reiterate, it is important that this iron is solvent inaccessible so that it does not undergo oxidation, therefore preventing this geometry from happening. Because if that iron were to transition to the iron 3 state, the iron could no longer adopt this preferred octahedral geometry and would therefore then be unable to interact with oxygen. So it's important that the heme group be highly solvent inaccessible. So the access of solvent to it should be highly restricted. And it is a consequence of the overall 3-dimensional arrangement of myoglobin that allows for that restriction of solvent accessibility. Okay, myoglobin is somewhat atypical in that it is considered to be an all alpha helical protein. If we look at some approximate proportions of alpha helix and beta conformation in single chain proteins, those are proteins that are built from a single polypeptide, you'll see that the percent alpha helix versus beta conformation it's fairly diverse. If we look at myoglobin, it has 153 amino acids present. 70 to 80% is going to be in the alpha helix conformation. Depends on the organism. In sperm whale myoglobin, it's about 70%. In others, it's greater, but there's no beta whatsoever. But then there are proteins like, say, cytochrome C, which has 104 amino acids. It's only 39% alpha helical, and the rest is in, well, not the beta conformation. It's random coil. So just because you have less alpha helical conformation doesn't mean that is offset by more beta. It could just be more random coil or other stuff. Then there are other proteins that are fairly evenly split. So ribonuclease has 26% alpha helical, 35% beta. To put it into perspective, here I am showing you the enzyme lysozyme. So lysozyme has both. You'll see most of it is alpha helical. If you look at the composition here on this slide, 40% alpha helical, 12% beta. But there's other stuff in that, so shown in pink, or salmon, if you really want to be technical, are random coil structures. And there's a little bit of beta, but mostly alpha helical. The enzyme chymotrypsin, on the other hand, if If you look at chymotrypsin, let me move these up so you can see. Chymotrypsin is 14% alpha helical, 45% beta. So you should see all of the beta here shown in magenta. There's a little bit of alpha helical shown in teal, but there's also a lot of random coil. So there's a lot of pink salmon connector structures in here. So this is just meant as a way to show you that globular proteins are going to be very diverse with regards to their secondary structure composition. You should not expect that they're going to always be all alpha helical or 50-50 alpha helical. They're going to be pretty broad and fairly diverse in terms of that composition. So why does that matter? Well, having diverse and varied secondary structural compositions matters because what happens is those secondary structure features will interact with other secondary structure features to start to fold. And by fold, I mean we start to adopt that final biologically relevant three-dimensional structure. So in order to do that, what we're going to do is basically take secondary structure features and start tethering them together and facilitating interactions that can be built into the overall three-dimensional globular protein structure. So this is going to begin at the level of the motif or the fold. So these are two terms that are used interchangeably. So a motif or fold is a recognizable folding pattern involving two or more elements of secondary structure and the connections between them. Okay, so let me put this into other words. Let's actually look at examples here. So if we look at, for example, panel A, the beta alpha beta motif or fold, what you're going to see is a beta strand that is connected to another beta strand by an alpha helix. And this is important because these beta strands are parallel to one another. So beta strand, connection, this is actually something of a crossover connection, but here it's in the form of an alpha helix, and that feeds right into another beta strand. So this is a beta alpha beta motif. So here, inevitably, there's going to be some interaction, non-covalent interaction between this alpha helix and each of these individual beta strands that sort of hold them together. But also they are just simply covalently tethered by the alpha helix. There are also examples of things called beta hairpins. So if you have two anti-parallel beta strands, often you'll see one of these is going to feed directly into the other via an arrangement that is referred to as a beta hairpin. By hairpin, this is, you've all heard of hairpin turns. These involve sudden changes in direction. So here, the beta hairpin involves a 180-degree change in direction. And so that's what you're seeing here. The beta hairpin is usually going to involve no more than 4 or 5 amino acids. And these beta hairpin motifs can be very, very important for interactions of proteins with their substrates. Enzymes, proteins that interact with DNA often have very positive charges, say lysines or arginines on the end of these beta hairpins that allow for interaction with DNA. There are other motifs like the alpha-alpha motif, or I love this one, the Greek key motif. In the Greek key motif, you have 4 beta strands that in primary sequence would be laid out like this, but they actually fold to give something like this, which is consistent with the Greek key motif. If you look at Greek art, you'll see this motif show up, and kind of looks like this. So to really draw this home, the term a motif or a fold, these, remember, are used interchangeably. These are just recognizable protein folding patterns that involve 2 or more elements of secondary structure and their connections. So motifs, folds, these are used to build the overall protein structure. In most cases, your protein structure is going to be built from multiple motifs, but in some cases, you may see more complicated motifs that comprise the entire structure of a protein. So here, as an example, this is the beta barrel motif. And it is very common to see fluorescent proteins adopt a beta barrel structure, and here the beta barrel would represent both a motif, but that motif also describes the three-dimensional structure of the overall protein. So to say this again, in most cases, you're going to have multiple motifs that build into the overall three-dimensional structure. In some cases, you do have a single large motif that describes the entire protein. And in fact, to build even further, sometimes the larger motifs will be built from smaller ones. So here you have the example of the alpha-beta barrel. Here you've got both alpha and beta secondary structures, and they come together to form, if you look at it from its side, something of a barrel, hence the alpha-beta barrel name. But the alpha-beta barrel is built from smaller repetitions of the beta-alpha-beta loop motif. So here we have beta, there's a loop that feeds into an alpha helix, which feeds into another beta strand. So this is a beta-alpha-beta loop. The alpha-beta barrel is built from beta-alpha-beta loops. So you should see here that motifs really usually you'll have to look to see what they are built from. Sometimes they are just large single motifs. In other cases, you have larger motifs that are built from smaller ones. The next layer of structure that we need to talk about is that of the domain. So a domain, technically defined, is part of the polypeptide chain that is independently stable or could undergo movements as a single entity with respect to the entire protein. So you're going to see that domains are built from smaller motifs, but the thing that really defines a domain as a domain is if I chopped it off from the rest of my protein, could it exist and be functional independent of the other parts of the protein? So here I'm showing you an example. This is a protein that has 3 domains. This protein actually is involved in DNA binding. It's a helicase. Don't worry about what that means exactly just yet, but here if you look at this, I've highlighted the 3 independent domains. So I know if I chop off at about this point, this green domain, just if I were to prepare and isolate a pure sample of just that green domain, I know that that domain is catalytically functional. In fact, I know this because I've done this. I've made this protein, I've made this domain. This is an ATP binding and hydrolysis domain. So if I make this thing alone, by itself, without the pink or blue domains, that ATP binding hydrolysis domain, it still catalyzes ATP hydrolysis. That's cool, okay. So this thing is tethered to a pink domain here. This is actually a DNA binding domain. If I make this all by itself, I chop off green and blue, this domain still binds DNA. The blue domain here is connected to the pink domain. If you purify, if you basically express this thing, chop it off right about here, it still folds and still does what it normally does. There's not an exact function that is associated with that domain, but the point here is simply that these are considered to be 3 domains because when you independently prepare them, you chop off the rest of the stuff, use genetic methods, and then express protein that is only one of these domains in isolation without the others, they still function independent of the others. They don't require the others. Now, if there's not a catalytic function, we would probably still call something a domain because it adopts 3-dimensional structure independent of the other parts of the protein. So here, this blue domain, even though there's not a catalytic function, it still adopts what is referred to as an alpha helical bundle overall 3-dimensional structure. So the point here is that these domains are not integrated into one another such that you need both parts to adopt that 3-dimensional structure. They can still become structured independently. So this is really a pretty cool aspect of proteins because often you're going to find that proteins are highly modular. you can think about this kind of like, a Lego set or, well, a car for that matter. If you think about each of these domains as having some function, you take two domains that, by themselves, they have unique function, and tether them together, you may get some new function. So just to give you an example, here you have an ATPase domain and a DNA binding domain. This is important because what happens is the DNA binding domain will bind to double-strand DNA, and with the help of the ATP hydrolysis domain, will actually separate double-strand DNA into single-strand DNA, which is a function that neither of these two domains could do on their own. So proteins are modular, and you can basically tether the parts together and design some new function, which is pretty cool. The recognition that protein secondary structural content is going to be widely varied has led to these development of classification systems based on that alpha beta content. So you're going to see proteins generally classified into 4 groupings based on how secondary structure is set up. So in some cases, you'll see all alpha proteins. These are proteins that have, when we talk about secondary structure, they are all alpha helical in nature. Myoglobin was a very good example of this. All of these have their alpha helical secondary structure, and of course they have the connectors between them, but it's largely alpha helical secondary structure. You've also got your all beta proteins. Same thing, except now it's primarily the beta conformation, whether it be beta strand, beta sheet, beta of some sort. Now, it becomes a little bit more hairy when you have proteins that have both alpha and beta secondary structures. So there is a distinct question that has to be asked. When we look at a structure of a protein, are the alpha and beta secondary structures kind of integrated together, or are they separate in their existence? So if we look at the alpha dash beta protein family, you'll see these secondary structures are pretty well integrated in alongside one another. So you couldn't easily take your beta content out without in some way harming the structure of your alpha helical stuff. So like if you look at this one, you'll see they're pretty well integrated together. In contrast, you have alpha plus beta proteins where the alpha and beta secondary structural components are kind of separate from one another. So if you look at this example, RuVe, you'll see there's some beta and there's some alpha, but all of your alpha stuff is sort of connected to your beta via this linker, and you could theoretically cut that linker and separate alpha from beta. The same is true for ribonuclease H, where you actually have a beta layer that has an alpha layer on top of it. So the point here is that though it may not always be easy to see, things are classified as alpha plus beta when there's less integration between the alpha and the beta secondary structural components. Now we can also characterize alpha beta content using the Ramachandran plot. So remember, the Ramachandran plot is simply an XY coordinate system plotting the dihedral angles surrounding each C alpha carbon. So we're looking at phi versus psi, shown here on the y and x axes. And if you remember, all these are rotations about 2 planes. So you've got, remember, each of your polypeptide, your peptide bonds can be treated as a plane. So you're just looking at the angle between 2 planes. That's all a dihedral angle is. Now, when we talk about characterization of alpha beta content, your dihedral angles that correspond to a beta conformation are going to very predictably cluster in the second quadrant here with very, very high predictability. So anti-parallel and anti-parallel and parallel beta sheets are always going to cluster up here in the second quadrant. Your alpha helix is going to show up in your third quadrant down here. So if you look at clustering, you should see that if there are a lot more data points, coordinates up here in your second quadrant, you're going to have more beta content than alpha. If you look at this example, you'll see a protein that is largely a beta sheet with a little bit of alpha helical stuff in here. That's, this is the Ramachandran plot that goes along with that. That you see more beta than alpha. But here we have an all alpha protein, and you see clustering in the third quadrant, which is consistent with the ability of the Ramachandran plot to separate alpha from beta. If you wanted to, you could calculate how many data points show up in the third quadrant relative to second and calculate percent secondary structure too, if you wanted to. To throw another example at you, there's a protein called monolin. So monolin is a protein that is responsible for giving certain materials their sweet taste. If you look at certain fruits, monolin is a protein that is perceived as being very sweet in taste. In fact, it's sweeter than table sugar, it's sweeter than many other artificial sweeteners that we have today. Now, monolin's also interesting because it has been synthetically prepared. So you can synthetically, basically using chemical synthesis methods, synthesize monolin and have it adopt its three-dimensional structure. If we look at monolin, you'll see there's a single alpha helix shown here, and the rest of it is the beta conformation. If we calculate a Ramachandran plot, you're going to see that there's some alpha helix, and the rest is beta. Now, this is cool because monolin has, we can synthesize it, the L and the D forms have been prepared. So L-monolin was prepared using L amino acids, D-monolin with D amino acids. And everything changes. If we make this protein with D amino acids, you will see that there's an inversion of all of your coordinates. So in your data points that show up, say, up here, where you have a Y value that's positive and an X that is negative, in D-monolin wind up down here with your signs inverted. So all of this winds up down here. If you look at the alpha helical components, your right-handed helix made from L amino acids would have coordinates that are both negative, so negative, negative, and in the left-handed helix built from D amino acids, that those coordinates just move so that they become both positive. So my point in showing you this is not only can the Ramachandran plot be used to characterize alpha beta content, but it can tell you in some cases whether or not you have a right-handed versus left-handed helix too. Remember, the left-handed alpha helix is built from D amino acids, which are not common in nature. They are not the norm. So this should raise some red flags to you, but it is possible due to a left-handed helix. So all of this was meant to show you that the Ramachandran plot is a very good tool to characterize not only alpha beta content, but the handedness of certain secondary structural features as well. Now we arrive at what I would consider to be a core truth of biochemistry. And that truth is structure dictates function. So when we talk about protein enzyme function, that can only happen if there's some structure to make that possible. So if we talk about an enzyme, you have to have a fully structured active site that positions all of your catalytic elements to interact with a bound substrate. That cannot happen if you do not have a structured enzyme. So structure dictates function. Now, this is interesting because what this has turned into is often people will look at the structure of an enzyme and assume that other proteins that look like it have the same function. So they say that proteins of similar structure should share a common function. But this is not always true. Evolution has made it such that your domains, they look like they look, they look as they do, but sometimes you'll see active site elements mutated so that they have different functions. So you will see examples here, in fact, that demonstrate that. So here we have 3 different proteins. They have the same domain type, so these all look about the same. They're considered to be the same domain type. They have different functions. 3 proteins, same domain type, but 3 different functions. They catalyze 3 different reactions. That is very interesting because here, what that means is that the similar structure does not lead to a common function. And this is a consequence of evolution. We have evolutionarily diverged, and that's what you're seeing here. There are other cases where you have same function, but radically different structures. So again, if we go back to evolution, we have here a case of convergent evolution such that evolution probably sampled different structures that led to the most efficient chemical path to do whatever it is these enzymes do. So evolution is usually looking to find the most efficient chemical path, but not necessarily, this doesn't require the same structure. So it's all about the actual output. Nature doesn't really care how we get there, just that we do so that we can survive. So here, let me reiterate this one more time. Proteins of similar structure do not necessarily share a common function, but structure does dictate function. I will go to my grave with that. Structure dictates function. So up to this point, we have been talking about protein structure through the lens of tertiary structure, and really we've been focusing on single polypeptide chain proteins. In those cases, your quaternary structural description is the same as your tertiary structure. They're monomers, so you don't really have to think about additional proteins or polypeptide chains being present. Now, in many cases, protein assemblies form such that you have a structure that is dictated by contributions from multiple polypeptide chains. This is protein quaternary structure. So many proteins have multiple polypeptide chains that come together. They interact via non-covalent interactions to give what is commonly referred to as a multimer, or in some cases an oligomer. These different oligomeric states are the quaternary structural description that we talk about. So if you have, say, 2 protein chains that come together, that's a dimer. If you have 3 protein chains come together, that's a trimer. In this case, this is actually the protein hemoglobin, which has 4 distinct polypeptide chains. It is a tetramer. So each individual polypeptide chain is referred to as a subunit or a protomer, but they all come together to interact and give the functional protein complex. So when we look at the structure of hemoglobin here, you can provide a tertiary structural description of each individual subunit, but you can also describe the overall structure in terms of quaternary protein structure. Okay, remember, structure dictates function. Now that I have reminded you of that, let me turn to a grouping of proteins that has challenged how we view that statement. So beginning in the 2000s and continuing to today, so this should tell you this is a fairly recent field, the observation of a family of proteins that are referred to as intrinsically disordered proteins has led to sort of our view of the structure-function relationship changing. So intrinsically disordered proteins are proteins that have properties that are very distinct from classical structured proteins. So they don't, on their own, have a core that is nonpolar. They are not necessarily globular. They lack this hydrophobic core. They're characterized by high densities of charged amino acids, such that they have a lot of lysines, arginines, glutamic acids. They also have a lot of prolines. But they also, let me just show you one of these things moving around. You'll see there is a core folded region, but that is flanked by these unstructured regions. So this is what we would consider an intrinsically disordered protein, because on its own, the ends have no structure, and yet these ends are what dictate catalysis. I know that's very strange, right? I just told you that active sites, enzyme active sites, have to be structured, and yet the parts of this protein involved in catalysis are not structured. So inevitably, what this led to was the dogma that structure dictates function. This was challenged. We had to think about how this could be. Well, I'll go ahead and give you the punchline. Through a lot of work, the field of structural biology has very intensively looked at intrinsically disordered proteins and has ultimately concluded that the dynamic conformational nature of these proteins. When I say that, what I mean is, these proteins are, each of these states, this thing is basically flopping about. Each of the states that contribute to the flopping of this thing are distinct conformations. And so this thing, these intrinsically disordered proteins are conformationally dynamic. But it is the dynamic nature of these proteins that make them polyfunctional. Okay, so here's an example. This is a protein that has a folded core. So we have a folded core, but the N and C termini of this protein are considered to be intrinsically disordered domains. If we look at a predictor, so you'll see this is what we call a Ponder score. And this is Ponder score as a function of amino acid position. The higher the number, the more likely that your amino acid at that position is going to be disordered. And by disorder, I mean intrinsically disordered. So if you look at this, you'll see here we start out at position 0, very high Ponder score. That would be my N-terminus, which I just told you is intrinsically disordered. And then you sort of feed into an ordered domain, come back up. Oh, there's another intrinsically disordered domain. Fall back down. A lot of this part is ordered. That corresponds to my core of the protein. But then at the C-terminus, I go back up and I get more disorder. So here I can calculate what part of my protein should be disordered, what should not. Now, why are my N and C termini intrinsically disordered in this case? Well, this is important because what actually happens is if you look at structures of this protein on complex with cyclin A or sirtuin or this CBP bromodomain or this thing here, you'll see that the C-terminal disorder changes. In fact, if you look at the coloring here, you should see orange corresponds to the cyclin A combination, green with sirtuin, yellow with the bromodomain, purple with this thing. The disorder goes down. So the C-terminus in this case becomes ordered. It adopts structure in response to the interaction with each of these proteins individually. That's important because ultimately that structure leads to different functions. So intrinsically disordered proteins are on their own largely disordered, but they adopt structure in response to interactions with different proteins. And these structures are different. They lead to different catalytic outputs. So intrinsically disordered proteins are referred to as being promiscuous because they interact with a lot of different interaction partners and they are involved in a lot of different functions that are the consequence of these different structures that intrinsically disordered proteins adopt in response to that interaction. There's another cool feature of intrinsically disordered proteins as well, and it is that they actually undergo something that's referred to as liquid-liquid phase separation. This is something that until about 10 or 15 years ago was just crazy. The idea that you would see phase transitions in the cell, that was something that really took a lot of convincing to establish. It was something that many people did not believe because ultimately what you're talking about in this case, this liquid-liquid phase transition, is the formation of an emulsion inside of the cell. Now this is important. If you think back to the hydrophobic effect, imagine you have nonpolar molecules that are individually hydrated. The concentration in a local space, a more restricted space of your nonpolar molecules is decreased relative to the situation where they cluster together. When your nonpolar stuff clusters together, you get higher local concentrations of your nonpolar molecule, because if you think about the volume occupied by that cluster, you're going to see more molecules per volume when they cluster relative to when they don't. So the same is true here. What happens is these intrinsically disordered proteins form these emulsions where the concentration of protein within is much higher than in surrounding space. This is really kind of cool because what this does is it leads to local increases in protein concentration that are necessary to assemble protein complexes or make chemical reactions go faster. So remember, in chemical kinetics, if we increase the concentration of a reactant, you're going to see faster reactions. If you have, say, a protein interacting with another protein, and say they have individually very weak equilibrium constants describing that association, if you increase the concentration of those components, you're going to make it more likely that they're going to associate. Okay, so if we look here, I'm just going to show you an example. We have this protein SPOP and another protein called DAX. These proteins are represented as being green or red. We can observe them because they are genetic fusions. So SPOP is down here. SPOP has been made as a fusion with this fluorescent protein M cherry. DAX has been fused to GFP, and you can see on their own there's these red spots, green spots. But over time, they actually come together and merge and turn yellow. If you look at the time dependence, you'll see two different spots that ultimately fuse together to give you something that is on the order of about 2 microns wide. So this is pretty cool because this is something that doesn't happen for proteins that don't engage in these liquid-liquid phase separation processes. So this is something that is specific to intrinsically disordered proteins. The why behind it is a little bit less clear, but the fact that you see local concentration increases in proteins as well as key reactants is really important because you ultimately form these non-membrane bound compartments that can do faster catalysis. So that you see increases in function in response to this phase separation. Now we have covered all that we are going to say about protein tertiary and quaternary structure. Next time, in part 6, we are going to turn our attention to protein stability and folding. That is, what factors contribute to the overall stability of a protein structure, and how do we get there? How do we go from being a newly synthesized, flexible, unstructured polypeptide into the structured proteins that we have seen here? So that'll be what we talk about next time.